Turns out, cancer combination therapies script. You've just been prescribed 2 cancer drugs. Together they work. But here's the problem nobody wants to admit. The pharmaceutical company has no idea which one is actually saving your life. It might be drug A, it might be drug B, or they might be doing something weird together that neither does alone. And right now, we have almost no rigorous way to figure it out. So when the FDA approves these combinations, they're kind of approving a mystery. How did we get here, and what happens when we try to solve it? For decades, cancer research operated under a beautifully simple assumption. If one drug is good, 2 drugs must be better. And you know what? Often they are. Combination therapies have genuinely revolutionized outcomes for people with cancer. We're talking about extending lives by months or years. That's not nothing. That's everything to the people living those extra months. But here's where it gets messy. When you give someone drug A plus drug B, and they get better, you're left with a fundamental question of attribution. Did A do the heavy lifting? Did B? Did they somehow synergize in a way that's greater than the sum of their parts? Before this research, we didn't have a great answer. We still don't, but now we're finally having an honest conversation about why. This study came from an unusual coalition. You've got researchers from Friends of Cancer Research, which is this non-profit think tank, working alongside scientists from basically every major pharmaceutical company you've heard of: Johnson & Johnson, Pfizer, GSK, AstraZeneca, Genentech. The lead author is H. Andrews, and this work pulled together perspectives from cancer centers like Dana-Farber and Fred Hutchinson, plus independent patient advocates. It was published in June 2026 in the Oncologist. This wasn't some academic exercise. This was the industry and the researchers saying, we have a problem, and we need to fix how we design these trials going forward. Here's what they did. They didn't run a brand new massive study. Instead, they reviewed existing trial designs for combination cancer therapies and analyzed them through a specific lens. How well did these trials actually establish the contribution of each drug? Think of it like analyzing cooking competitions. You've got a dish that tastes amazing, but you're trying to figure out whether it's amazing because of the sauce, the protein, the technique, or some magical combination of all 3. The traditional gold standard is a factorial design trial. That's when you test every possible combination: drug A alone, drug B alone, drug A plus B, and placebo. But here's the catch. In cancer care, that's often impossible. If you have an aggressive cancer, you can't ethically give someone a placebo when a known drug exists. If the disease is rare, you don't have enough patients. If treatment standards are evolving rapidly, your trial design becomes obsolete mid-study. These researchers looked at what alternatives actually exist and how well they work. What they found is both obvious and uncomfortable once you hear it. Most combination trials can't actually establish individual drug contributions with any real rigor. Instead, they test the combo as a black box. The trial shows that A plus B beats the control, but the FDA has no mechanism to determine whether it's 80% A and 20% B, or 30% A and 70% B, or some weird interactive effect. This matters enormously. If B is actually contributing almost nothing, you're exposing patients to extra toxicity for no benefit. If A is the real hero and B is just along for the ride, future research gets misdirected. The researchers identified several alternatives to factorial designs that could work better. Basket trials that follow individual drugs across multiple cancer types, adaptive designs that allow trials to shift focus mid-study, real-world data collection that tracks what actually happens when people take these combinations outside controlled settings. None of these are perfect. Each comes with trade-offs between scientific rigor, cost, and how quickly you can get results. But they're all better than the current system of approving combinations without actually understanding them. What does this actually mean for you? If you have cancer and your oncologist prescribes a combination therapy, there's a reasonable chance that neither your doctor nor the FDA has solid data on which drug is doing the work. That sounds alarming, and in some ways it is. But put it in context. These combinations still work. They still extend lives. The science isn't wrong, it's just incomplete. The real implication is that we're potentially leaving better outcomes on the table. If we knew exactly what each drug contributed, we could optimize dosing. We could reduce side effects by lowering the dose of a drug that's not really helping. We could predict who will respond best. Right now, we're using combinations that work, but we're not optimizing them. It's like having a car that runs fine, but never having looked under the hood. The honest limitation here is that implementing these better trial designs costs money and takes time, and the incentives don't always align. Pharmaceutical companies want to get products approved. Patients want treatment now. Regulators want both things to happen quickly. Rigorous factorial trials slow everything down. The research doesn't solve this tension, it just makes it visible. This work sits at an interesting intersection in cancer research. On one level, it's about methodology. On another, it's about regulation and economics, and how we translate research into practice. The field is moving toward more combination therapies because they work, especially in immunotherapy and targeted cancer treatment. But as combinations become standard, the problem of attribution gets worse, not better. When you have 3 drug combinations, or 4 drug combinations, the complexity explodes. The researchers are essentially saying we need to rebuild how we think about trial design before we reach a point where most cancer treatments are combinations and we understand none of them. The immediate next step is adoption. Will the FDA actually push for these alternative designs? Will pharmaceutical companies willingly make trials longer and more expensive? The bigger question is whether we're willing to slow down short-term approval timelines to get better long-term understanding. That's ultimately a values question, not a scientific one. The research suggests we have tools to do better. Whether we use them depends on priorities outside the lab. Here's what sticks with you from this. We've been approving cancer combination therapies without actually understanding how they work. We know they work, but that's different. It's like approving a bridge because traffic moves across it without structural engineers ever examining what's holding it up. This study is the moment when people in the field said out loud that this was a problem and that we could fix it. But fixing it means making harder choices about speed versus understanding, cost versus knowledge, helping today's patients versus learning from them better. Turns out, scientists have been busy, and they're not done yet.